Welcome to everybody who decided to join us for seminar today. We have this talk will be in two places at the same time. It will be at the Sano and on the third floor, and as well it will be also online. So, well, we will start in a second. There are still many discussions here.
Yes, about food special.
But there is time for discussion when we will, after the talk.
Yes.
And I hope that it will be also possible to, as usual, to ask very short questions which are required to understand the talk. But we will have a serious discussion at the end of seminar. Today we have with us Professor Joel Arrais from University of Coimbra, from Department of Informatics Engineering. And well, I think that Joseph, the leader of one of Sano team, knows probably much more than me about Joel. So, Joseph, could you be so good and introduce our today's speaker?
Yeah, so no worries. Thank you, Marian, for that. So thank you for the nice presentation. First of all, Joel, thank you for being here. So, but you are seeing snow, so so that's our present to you. So we are giving you snow to enjoy the snow. Yeah, that's what we did.
It's possible to take without paying taxes.
Oh yeah, that's even better. That is even better if you don't pay taxes on snow. So Joel is associate professor at the University of Coimbra. She's leading a team there that is working on exploring diverse type of technology applied to drug repurposing. So I think he's going to walk us through that process and do some of the experiments that Joel is running and some of the results that he's been achieving. So he's also part of CESUC. So CESUC is one of the research centers at the University of Coimbra dedicated to computational science and informatics. He's also supervising or co-supervising the Joseph, so one of my PhD students. And I met Joel from that perspective, but then we started to develop a closer collaboration towards different areas. So today, Joel is going to walk us through what the things he has been doing, and in the end, we open the floor for questions. And also, if there are people online that want to come to Sano, we still have lots of pizza to share. So if you are close by, please come, and then we share the pizza. Okay, Joel, the screen is yours, as Marian says normally.
So now,
and the floor, yeah, sorry, the two things are yours.
So first of all, thank you so much for the invitation. Thank you for hosting me, Jose, and also for Sano for having me for this week and a half. And also, thank you for the pizza. I think that it was really, really very important, as I was able to understand, to make sure that all of you were here. Okay, so the goal that I have, it is to present some of the work that I have been doing in the lab in Coimbra in Portugal over the last years, but also, and as much important as that, to try to bring the discussion how some of the things that we are doing could be connected to some of the things that you are doing in Sano in many of the different groups. Okay, so more important than the things that I'm going to present here are the ideas that we can explore further in the future. So I'm not sure if you know the University of Coimbra. So it's one of the oldest universities in Europe. It was founded in 1290. So, and it is one of the biggest universities in Portugal. And if you don't know it, you are mostly welcome to visit and to pay me a visit. And I will be very glad to have you there. And also, I'm associate professor at the University of Coimbra in the department informatics department, and also researcher in the CESUC. But my lab it is in artificial neural networks, and it was created before artificial neural networks were fancy as they are now. So it was created in the 90s. and we are proudly keeping the name. And my research interests are in applying AI and computational models to biological problems. So especially for drug generative models, so for being able to generate models from de novo or for repurposing, of course, drug target prediction, and also genetic variance analysis. So you're analyzing QAS or exome or genome data sets, and also for pharmacogenomics. Okay, so I'm not. It is not the goal for my talk here to show in detail everything that we are doing. Okay, so I just want to point you to the main ideas and to the vision that we are doing in the lab. And I think that the first thing, it is actually something that all of you must already be aware, because we share the same background, that coming from computer science or from physics, and trying to promote this to biology, and that it is that the research in biology, in fundamental biology, it's moving away from, and the need for computer science working with biology, it's moving away from the experiments, it's finished, and now the bioinformatician or the statistician, it's going to start to analyze the data. So this would be something like in a post-mortem analysis of the data. I think that this is completely over. And I think that the shift, the paradigm now, it is that we are the ones that design the experiment that actually use previous knowledge and data to create the main hypothesis. And then the lab, very important, of course, but it is used to validate these candidates or these hypotheses. Okay, and this seems like normal, I think, or should be for some of us, but this is something that it is a big paradigm shift in the way that the research in biology it is conducted. And actually, in my lab over the last decade too, we have been trying to promote this and to show how this can be implemented. Okay, so instead of just going in the end, the statistician or the bioinformatics analyzing the data, we start from the beginning, and we are the ones that create the hypothesis. How we do that. So actually, I think that this is possible now because there are plenty of data available from omics studies and from many other kinds of data, and also unstructured data like PubMed from publications, but also because all of the advances in artificial intelligence that enable to extract data and knowledge from previous published information or from public studies. Okay, and so most of the things that we are doing in the lab, it is actually to demonstrate that this is possible and that this are already happening, and especially we are applying this for the drug discovery pipeline. I'm not sure that you are familiar with this, and I don't want to get into many details, but I think that the main pointer, it is actually that the drug discovery pipeline is a very complex, as one may imagine, but in the first 3 stages of this process, there is the target identifications, and then the lead discovery, and then trying to promote this to a possible candidate. This is mainly a data-driven process, and it is a process where the use of computers and some of the things that they are quite normal for us, they can help a lot. And despite that, things are changing over the last years, but until now, the classic, even computational classic methods were the king on this approach. And what was the result? A lot of candidates would fail. Most of the candidate drugs would fail on the later stages. So the vision that we have, it is to try to promote, like this paradigm shift in research, and try to promote this for the drug discovery pipeline for the initial stages, and it is to be able to design the drugs, and at the same time that we are designing them, we are already validating them, okay? And so this is one of the things that we are trying to push, and the pharma companies, they are mostly well involved on this. So actually, and conceptually, the things that we are doing, it is from omic studies and omic data that is available, that it can be gene sequencing, but also transcriptomics or proteomic studies. We are able to simulate in silico the relations that are happening in order to find the candidates, so the best targets for a given disease or for the problem under study. And then based on that, we have this generative model that it can be based on reinforcement learning or based on agents, or it can be with human in the loop that at the same time is able to generate candidate molecules, and it is able to evaluate them. And then we have already tried to synthesize these molecules and to validate them with different levels of success. Okay, so the first example that I have on how this can happen, it is with this time-dependent study. And okay, so and what we have, it is in this specific case, we have worked with gene expression data, so transcriptomic data, where we were able to find what were the genes that were differentially expressed, in this case for a time series of 24 hours, for patients or for cells that were infected with HIV. And then we were able to get the transcriptomic response, and then we use it to build a regulatory model that would be able to explain why some genes would get differentially expressed based on the observations that we had in previous time points. So a very important point, it was that we started with data that was already publicly available, okay, from a study that was already analyzed. And then we were able to get to build this computational model that would explain and simulate everything that was happening at the transcriptomic level in the cells. And the way that it works, it is as an optimization problem where we have multiple targets involved. And the way that it ended up in the end, it was that we have created a computational model that it is able to simulate or to represent everything that was happening in the cells based on the transcriptomic analysis. Of course, that we have used protein interactions and all kinds of networks analysis, but that was the model that we were able to build and to obtain from this data. And now one would assume that this work would be completed. But then we wondered, what if we were able to take this model and to pose him some questions, okay? And the questions would be something like, what would be the proteins that I would have to block, to inhibit, and at which time point in order to stop the progression of the infection from the HIV, okay? And then what we got, it was this list of target proteins, and for each one of those and the respective phases, for each one of those, we bought the inhibitor. Well, not me, because I do not do wet lab work, but we bought the inhibitors, and we were able to validate with human cells what would be the response. And actually, it was interesting that we were able to find some possible candidates. So that we tried as a benchline the AZT, that it is the antiretroviral drug used for HIV patients that works quite well with early time points, but it doesn't work as well in later stages of the infection. But we were able to find possible candidates that were able to be possible. And the interesting thing is that these candidates, they were not identified in the initial published study. and they were here. This work was conducted while I was in the Carnegie Mellon in the US back in a couple of years ago. But I think that it illustrated quite well how we can start from published data, create computational models, and then use them to go to the lab in the last stage to validate this hypothesis. So the second example that I have here, it is now we have identified the target or one possible target for the molecules, and how can we validate it? So, meaning how can we design or identify molecules or new drug candidates that would bind to this target. So in this example, we have not worked specifically with the targets previously identified for HIV. And actually, we have a lot of work in drug repurposing or reusing some drugs that I do not have here in these slides. But in this case, what I have, it is we have created a model that it is able to generate de novo molecules with a specific aim. Okay, so I think that is quite easy to explain now, not when we started this 6 years ago, but I think that it's quite easy to understand now or to explain to you that this is a ChatGPT for molecules. Actually, at the moment, there are already pre-trained models that are way better than the ones that we have published in 2021. Okay, and that were trained in computers that we don't have access to, and so. But the concept was that we have built a language model, and we were able to show that it was able to learn the grammatical chemistry rules from the molecules, and we were able to generate molecules that were grammatically valid. Okay, and that was one of the first good moments that we have. And then we try to use this to many different things, but one of them, it was for the adenosine A2 receptor, because mainly because it was a very well studied receptor, and there was a lot of public data available, and they're still missing a good candidate for drug. And also, we wanted this molecule to also have to be able to permeate the blood-brain barrier. So we have these two objects at the same time. So I don't need to explain this, and I don't think that I also need to explain how deep reinforcement learning works. But either way, so actually, what we do we have this generator that actually it is our language model that we were able to train with millions of molecules that are available. And we also have this predictor that actually is not just one predictor, but it is a set of models that are able to evaluate each one of the individual molecules that we have for different properties or for different conditions. And actually what we do, it is to be able to join this reward in a single reward, and to be able to use this in a Markov decision process, but to be able to iteratively be able to improve the training or the bias of the generator towards something of interest. Meaning, we start with something that is very general in the beginning, and then it tries to learn with the experience with the environment. And in this case, the environment, it is the rules that the predictor can give us. So I don't think that this is very important, but actually we try to model the molecules as vectors in this specific approach. And this was one of the most basic approach that we tried. So we model it as a language model, okay? And then we are able to train it in thousands of approach. Also, for the AI model, we started with LSTMs. I'm not sure if you are familiar with LSTMs, are you? Yeah, okay, so yeah, but actually they were quite popular a couple of years ago before transformers. Now everyone should be using transformers, and we are also using it. But I think that LSTMs, they are quite easy for me to explain how this works. because, yeah, I think that is way more intuitive. So we treat this as a sequence to sequence in a way that for each one of the characters, and the character, it is a chemical atom, we want to predict what it's going to be the next one, and then the next one. So it works in the same way as our phone. It is able to predict the next sentence or the next character that we are inserting while typing a message. So actually, there is in the same way that this works, and it is able to learn what is going to be the probability of the most probable atom in this. And for the property predictions, well, we can build anything at all. So we could build specific rules, okay, or we can have these more advanced things that we are using. That neural networks. Why neural networks? Well, that story of the hammer. When you know how to use the hammer. So we are in the artificial neural networks lab. So the neural networks, it is more easier than if-else for us. Okay, so but it could be a simple if-else if we know the rules, of course. So, but for us, this was quite easy to train for the biological affinity, and we can build them like in a fully connected layer like this one. But I think that the most important realization or conclusion that we got, it was that we were able in first place to create molecules that they were valid. Okay, this seems really obvious now, but at the time it was really important for us. And then that we were able to build predictors with the available data that would work, kinda. And then we were able to merge all of these things in a single setup, where we started with our unbiased generator. We create a duplicate of it, a replica, and then we use this loop of reinforcement loop and be able to use, in this case, multiple properties and then obtain the reward, okay? So in this specific case, we were able to show that we were able to get molecules that would be possible to synthesize, that would be valid, and that's using computational models like docking and others that may work. We were not being able to synthesize them specifically, but we were able to get all of these molecules that actually would also already be available, similar to molecules that we do not use for training, but that we knew that were already relevant for this for these targets. Okay. And this was, like a cornerstone for us, because it shows that we can use these principles to other problems. And that was the thing that we have done after that. So I think that one of the obvious things that we have done after it was to use this for to improve the model for transformers, because they would be easier or the results would be better to implement. So we write everything for transformers. This was already back in 2023, I reckon. And we were able to rewrite everything for transformers. And also we were able to get a major improvement. And in that specific case, we were still not synthesizing it, but we were getting with better confidence. And then we took all of that confidence and we went to someone that is a chemical synthesis working in the lab, and we showed them our molecules. So actually, I think, I don't have them here, but we showed them our models, and they just told us that all of them are ugly, very ugly. I would never synthesize them because they are very, very ugly. And we were, like, you guys are on research, that feeling. Then you are full of expectations, and everything really goes down because all of your hope is destroyed by someone that knows how things work. We have already, all of us, been there. So what we have done, retirement would be an option, but then we thought that, okay, so let's try to retrain the model with this information, try to understand why it failed, okay, and try to improve it. So, and we have done that once and twice, and eventually the molecule started to, to look better. I don't know what, but and we were able to finally be able to synthesize them or to convince them to waste money because we were not paying. So to spend money to synthesize them and to test them, in vivo. And the results were interesting. Okay, and then that was the first approach that opened the opportunity, to show us that some of the works that we are doing, they can have a possibility to be used in something real. And then something really bad happened. That it was that a lot of my PhD students, they were hired by the big pharma, because the work was, being so promising, and at least two of my PhD students, former PhD students, they were hired for working the big pharma in establishing AI for drug discovery, there.
Excuse me.
Yeah.
What do you mean that they were ugly at the beginning?
The molecules.
Yeah, the molecules were ugly. Like
So I cannot explain.
Okay.
When I hear that the first time, it didn't seem like a good thing, but it was not synthesizable.
Okay.
That would not be easy to construct,
Okay.
and that would, even if it was possible to synthesize, there were parts there that would enable it to work properly.
Okay. So from the biological perspective, they were
everything
valid.
everything.
Okay.
So
Yes, can sort of view sometimes they already know by experience that it will
Yeah.
not possible to decide them or that it will not work. So they just have like the impression that it's not possible even to prepare such types of drugs.
Okay.
Yes.
But it's like a gut feeling.
Yeah.
Okay.
well, it's the gut feeling of someone that has the experience of a lot of failures. That would be really nice to take our the brain of the person, and but that's not legal.
And then with some training, like additional training, you can do.
Yeah. So actually, we went step by step, and we tried to understand why is it failing, or
Okay,
why do you
okay.
think? So we tried to account for other parameters, and yeah. So, and then we were able to synthesize them. So, and of course, then we are still applying it to other goals right now, and we are trying to synthesize them. And it is a specific line of research that we have. Actually, so in this case, we are just applying this to small molecules, but we are also applying it to peptides therapies. Not sure. So it is something a little bit different. To peptides.
That lady over there knows a lot about that.
Yeah, so we were able to retrain all of the model to peptide therapies, so to peptides, and we were able to generate peptides for a specific goal. So we started with something very simple, that it was antimicrobial properties, and we were able to, to get some sequence, and we were able to validate them in silico, and we are applying, I don't know, for a patent with our colleagues from Macau. Okay, but but the thing that I really enjoy to do, it is to start from these models and try to bring, all of this to the people, to the researchers in the lab. Then, and those are the things that I control, and that can. One of the other lines of research that we are doing, it is, I'm not sure if you are familiar with variation autoencoders. Are you? Okay, it's good to see some heads, but in that
The others are shy. The others are
shy. Because so variation autoencoders are very, very interesting, and I think that it is easier to understand with this picture. And one of the things is that it can obtain a compact representation of, in this case, 2 dimensions of a sample. Okay, so a sample, like in this case, that has I don't know how many bits for each one of the images, can be represented in a 2D dimension. But the thing that is really important, so it is something like a PCA so far. Okay, do you know PCA for sure? But the thing it is that the space in here is regularized, meaning that we can select any specific spot in here and go backwards and generate an image that would correspond to that position. Okay, so we can sample, we can generate new instance. Okay, and this is being widely used in some of the image generative models. Okay, not now because now everyone is using diffusion and stable diffusion, but you get the concept. So actually, what we have done with this, it was to use TCGA, that is a database for gene expression transcriptomic studies from cancer with drug-related information. And we were able to get a compact representation, not 2 dimensions, but like 10 or 100 dimensions that would represent each one of the genetic transcriptomic profiles for each one of the patients and their response to a given drug. And we were able to create a model that was able to simulate the response to new drugs. Okay, does that make sense? So we trained it in previous information, and then we were able to get new samples that would get the transcriptomic response for a given drug. And then we took that box and we couple it with the generative model that we already had. And for this generative model, what we were doing, it was to generate new instance, okay, new molecules. So, and now we want new molecules that would act to reduce the expression, to bring the expression from a patient with cancer to the basal level, to the normal level. And that would be something of a reward function. And then the molecules that are being generated are the ones that are going to minimize this response. So we are designing de novo, and we are obtaining these genetic profiles, and the objective function would be to minimize this. Okay, so and after this, so we'll just let the computer running, and then it will get us these genetic profiles to the position that we have, and then we get these molecules. I'm not a chemistry guy, sorry. And then we got these molecules that we were able to validate just computationally so far. But then we showed that it was possible to start from the expression profile and to take this. And now there are, this was 3 years ago that we have done this. And then there are, like tons of works in the literature that, of models that try to do this, and they show that they can do it better than us. But, like the idea was here, and I think that we were, I'm very proud that we were able to take this idea so far. So this work itself, it's not very important. Well, it was very important for us, but it is not very important for this discussion, because the main concept it was that we were able, attention is all you need. Everyone knows this paper, right? No?
Yeah.
Okay. So we were able to bring attention mechanisms to this model, and we were able to improve the performance. One other separate line of research that we have in the lab, it is to try to not merge the transcriptomic profiles themselves, but also try to merge the sequencing profiles. Okay, so what we were doing, it is try to understand how these genetic profiles would merge with the development of drugs. I know that if anyone is from here is already thinking on the SIPs, but we are trying to think outside of that, and we are trying to and one of the works that we have, one of the projects that we have, it is with the cisplatin, that is one of the drugs used for the treatment of multiple cancers, but it has a couple of side effects, and one of them it is the ototoxicity, so the loss of hearing. Okay, so what we are doing, and because we know that there are some genetic variations that would have implications in the odd ratio of the prevalence of the hearing loss, it is to try to create these polygenetic risk scores based on this information. But actually what we are doing is to go beyond the traditional polygenic risk scores that just make a linear correlation, and we are trying to make them as neural networks or nonlinear relations between these associations. And actually, this is a project that it has 8 partners from Portugal, Spain, France, and we aim to recruit 750 patients. I'm just the guy from the computer side, okay? So I will be mostly involved in these artificial intelligence models and in trying to propose them to the consortium, okay? So I'm getting very close to the ends, and I will be really open for discussion afterwards. But I just want to rewind and to make clear that the thing that we are trying to build, it is to use data that it is already available for everyone, and I think to build these computational models that are able to create hypotheses. Okay, and then based on this, we are able to generate new molecules. Okay, there are a lot of limitations in here. Okay, there are a lot of limitations, and one of those limitations, it is actually how do we include all the previous knowledge that is in structured, but also in an unstructured way. And that's why I really want to bring the discussion with Jose and Joseph to here, because as it is right now, we are simply not considering it. We are just
we locked Joseph for 2 hours in a room without letting him to leave.
Sorry,
we are discussing that.
And so how to bring this, and I think that the knowledge graphs and all of these representations of a more open world will be quite interesting to include in here. And also many other possibilities of improvement that I want to bring here, and I will be mostly open to everyone the discussion. And so, okay, so I don't know. Most of these photos are already updated. I'm quite sure that Jose, you know this guy.
Yes.
Sorry,
you should remove that one. You should
sorry.
remove that one.
I will.
You should take him.
But
He's going elsewhere.
Yeah, I don't know where.
Yeah, I don't know where.
So, but this actually, it's my team right now, but just the PhD students, but we are more or less 15 right now, with also a couple of master students, and also something really, really important, the backgrounds that they have. So some of them, they are from pharmaceutical AI, a PhD program in pharmaceutical AI. So some of them, they are from computational science, others are from biomedical, biochemistry, chemical engineering. So very complementary background that I try to bring together. Most of the times I'm the only guy from computer science, really, from the bachelor. So
I know the field.
So I think that, those were the things that the pointers that I wanted to start the discussion. And thank you so much for staying here. Even after the pizza,
Many thanks.
Yeah.
Well, you have at one point you have mentioned that it will be multi-criteria optimization. Is it really multi-criteria?
No, it is not.
Yeah.
It could have been, but we have not put efforts into that. But the problem itself, we have multiple criteria, but actually we are just cheating. We are just creating a formula that encapsulates the multiple criteria. We are not looking to the Pareto front or to something like that. But yeah.
So I understand that it is multi-criteria in that sense that some of these criteria are solved before you will go to, let's say, optimization using this gradient method or something like this, yes?
So sorry.
In selection of candidates for solution, you already use some criteria, yeah?
Yes.
And then you tuning this with.
Okay. So I think that I understand the question. How do we tune it? By exploratory analysis, actually. Yeah. So we look to the possibilities.
Okay. So it's So this multi-criteria is just for general, let's say. Well, okay, thank you.
I mean, it got me curious. Maybe it's like a little bit too specific. Maybe we should talk about it later. But it's always interesting when you said at the beginning the thing wasn't working with the molecule generation, so they're like ugly, as you said. It's like
It wasn't me who said that.
and I'm curious, like knowing some feedback from the chemistry specialist, I assume. How did you manage to incorporate that in actually the neural network design and training? Because that's not always obvious, because they might say, oh, this never goes together, but you can either include it in the architecture or in the loss, or there are many things that can be like as the control points of the neural network. So I know that's like a very detailed question.
No. Actually, it's a very specific and a very smart question. And and actually, I do not have one answer, because we have tried different approaches over the time. So over the time, we tried different approaches for actually to trying to answer that. Our initial approach, it was that we didn't want to put things on the reward function that would be static. So we didn't want to put conditions there. We wanted to show, because we wanted in one of the stages to publish this in a computational, more computational journal, we wanted to show that the model was able to learn based on the representations. So in one of the approaches, what we have done, it was, why is it ugly? And one of the answers was because this group wouldn't be, like relevant for this. So we tried to show examples to the model where this group would not lead to, to good results.
So like counter examples.
Yeah. Or meaning using the input that we have for the specific models and retrain the model with those examples. So without putting anything specifically. And actually, we were at a certain stage. There's one metric that is the how that defines how well it can be synthesized.
Okay.
But we were not using that as an approach for training the model, because we wanted to show that the model was able, that the molecules were synthesizable without having this specific. It's something that you do when you are in a closed box. And then at later stage, we just assumed, no, we are going to include this somehow in the molecule, like it was as part of the reward. And so I don't have one fixed question, because it is a process, and we are always trying different things. I'm not talking about some of the most recent work where we are using diffusion, stable diffusion. I'm not sure. Yeah, so it is one of the state-of-the-art models, and we, instead of using this as a language model, we are using the image map of the interface of the protein to try to design what would be a pharmacophore. This is so, but we use the image, and then we took a photo from the moon, and then we want something that would land there in a specific part. And then we take the image to build the thing that would couple there. Does that make sense? So it is the approach that we are doing now, still unpublished so far. But and then we build the concept of the drug, and then we go to generate the drug based on that, not based on the language model. But
Okay. I bet there's like any kind of publication about this. So later on we can have a look.
Yeah. But your question was really on the spot, and I really like that.
Well, we have Wiesław who is online with question. Wiesław, can you ask your question? I hope it will hear you.
Yes, can you hear me?
Yes, perfectly.
Okay, hello, thank you for your presentation. I'm not quite sure if I have the right question, but anyway, a few days ago, Mark Zuckerberg from Meta, and 2 days ago, I saw the first press statement announce open source general intelligence. Basically, he says that this is a new AI system which can reason across scientific domains, and he claims that this is your lab partner in your pocket for everyone on Earth. How do you think how this initiative will change your work and the work in your whole field?
Well, if it's coming from Mark Zuckerberg, and if it goes as well as Meta itself, there there's not much to expect. But now seriously, so the using agents and being able to build agents that help in the tasks is one of the things that all the companies that work in AI are interested, right? For all the tasks. I'm not the best person to discuss if it is going to be possible or what it's going to be possible. Okay, in the scope of drug discovery, there are also a couple of projects, specifically on that that try to build agentic AI for drug building. And one of the PhD students that is starting now, it's also working on that. In a more broader concept, if AGI is possible, so what are we doing here? Let's just go home and someone else will do all of our work. But I must say that I'm not the best person to discuss AGI or the impact that it will have in our lives. But specifically for drug discovery, there are a lot of research being done on the use of agents but one thing, in the end, it's not going to be much different from this, where we have different models for evaluation, and it's not most of them. I don't think that they are working in an open world. They are working in a quite of a well-defined world. Until I'm sure wrong, I don't know.
Okay.
Yeah.
Okay, thank you, Jel.
Many thanks. And now we have one. I hope that now we will be successful with Adam. Adam, can you go on?
Yeah. Can you hear me?
Yes.
Yes.
Yeah. I will be tomorrow, so I invite
Yes, you can come to the second floor and meet Joel if you want to have a chat.
Yes.
Yeah, of course. One caveat regarding the generative model on Smiles. Smiles has grammatical rule which gives you possibility to error, and this error are quite frequent. That's why what this string gives you is not chemically valid. It doesn't.
So you
exist.
sorry, you are referring to the stereochemistry.
Yeah. In general, it's quite well known that we are not using smiles in generative models. We are using selfies. This representation don't give you space for errors because it's like putting building blocks, and every sequence ordering of these building blocks always gives you the real structure. The problem with smiles is that like putting the dot or other question marks in the middle of the sentence, so the sentence lose the meaning. The same is here, like you are putting some marks which totally breaks. For example, here as you present, you have this CC and this other sign. So putting this in bad place results with errors. So that's why people are using selfies representation for this text, chemical text. But it's only caveats. But what was interesting for me, you mentioned about small peptides, right?
Yes.
And antimicrobial properties, right?
Yes.
So I also recommend the work from our colleague from AGH near Sano. and they used molecular fingerprints. It was quite surprising that this static bit representation outperformed all benchmark, because peptides are quite local. That's why it beats even Meta large language model SM2 or SM3 in domain of the small peptides, because yeah, the interaction there are quite local, and this work will be published already, or it is published in bioinformatics. So for you, if you want to publish some generative model, it's good to use it as a benchmark, because in most cases, molecular fingerprints are the best.
Oh my God, at least one person was able to understand my presentation.
And that's
Thank you so much
you have a second one over there. So that lady over there also understood. I didn't understand anything,
Not just was able to understand, that was able to, like,
to paint.
like, this is, does it hurt?
Yeah,
it hurts a lot. So first, smiles. Yeah, so smiles have that problem. Okay, so there are different ways to solve that. One of them, it is to try to use selfies, and we have done that. So we have tried all the different language representations, but selfies would have a lot of problems while training language models. Okay, so we were not able to use them as efficiently. Then we have to deal with the problem of chemistry. And then what we dealt with, it was, I cannot explain how, but we have created a model that creates all the possible combinations and then evaluate them separately. And then we have to filter in the end while generating the candidates. It's not a clean process, okay? And that was something that we have to, one of the problems that we have to solve. The approach that we are following now, it's for many different reasons, but one of them it is because of the limitations of language models, and one of them is that one. So yeah, very important. The second one regarding the peptides. So we do not use any kind of features. Okay, so we do not use, so it is an end-to-end deep learning model. So the input is the sequence. We do not use handcrafted features, and that's why we didn't want to use the fingerprints. And also, I think, and this is getting muddy. That's when you are using specific features, you are more prone to overfitting eventually. Okay, so that's why.
Or in this case,
I would say in general, but in this specific case, and the use of extended fingerprints is that the approach that we are doing, it is for generating. So we are using it to generate new instance, and yeah. But I understand your comments. And while we have published it, we compared with, at the moment, the reference models for prediction. Yeah. But your questions, I would be able to speak for not just 1 hour about all the steps for those, and the advantage and the disadvantage of each one of the things. So I really would glad to meet you later if possible.
Yeah. So definitely I will spot in second floor tomorrow.
I was wondering whether you tried to, or you saw some variability between different populations in terms of drugs applicability, whether, for example, because some diseases have a strong genetic foundation, therefore different populations may have different targets and want to address with a drug. I was wondering if you also did analysis like that in your work.
Mhm.
or if you are interested in having maybe a model like that, fine-tuned on specific populations to better adapt to
Mhm.
certain direct factors.
Yeah, so the goal, not specifically to this work, but to the personalized medicine and to the personalized drugs, it is to try to fit the genetic profile that can be a single profile or it can be the profile from a population to a specific drug. And we haven't done that, but that is something that I want to look forward to work in the future, and we are already doing some proposals that would in that.
Yeah, so with Joel, we have a proposal that was submitted by IBLI on AMD. So where Joel is going to work on the drug proposing part, and we are going to work on our models from the clinical and generating the features. So we are trying already to
Well, zero work then from my side.
yeah,
But-
probably, yeah,
But it is something that I want to push
Yeah.
the review. So it makes total sense. I think that you are discussing.
And also, I was curious whether you tried to use image analysis, computer vision. For example, translating the signals you have, time series you have to images, since you're using version of the photos, for example.
Mhm.
That could also be a direction to switch to the computer vision side, so to say, or even integrate two things together, maybe they cut different patterns. I was curious if it's something you thought about, considered.
Yeah. So I would say that the first more. So I have never worked with computer vision. Disclaimer. Full disclaimer I answered. But one of the most relevant deep learning methods for computer vision, I would say that were the convolutional neural networks. And it actually back in 2018, one of the PhD students was able to show that we were able to extract features and to build end-to-end just based from the sequence based on convolutional layers. And now everyone uses not CNNs, but convolutional layers everywhere. And I think that also we are using actually images for the proteins. We are taking photos of the proteins, and we are using these. Well, they are not photos; they are heat maps from the interface of the proteins, and we are using them to build pharmacophores. So that would be much there. And the methods that we are using are exactly mapped from computer vision, like diffusion and UNets, and all of the things that you can use. So yes.
In the case of time series, I think that the structure of peptides is changing too quickly. So it will be very difficult and it's very different than. So even when you are preparing this model for peptides or proteins, it's like 107.
But you do the analysis sometimes series, right?
Yes, like it's this model. Don't forget there are time series. You coverage the data from modeling and then you
I was not expecting this enthusiastic.
I took them yesterday to pizza. Two of them, they can be asking questions, They bring them very nice, so they should be interacting.
Because I know that I have made some of my homework, and I saw that some of you work in very different things. So, but I'm really impressed with the questions and the way that you.
This is just like our curiosity, but I remember that cells had this whole signaling cascades and they were like super complex. It's not computational, but do you just try to bind the molecule to one protein and that's it? Or do you consider the entire process of, because how much do we know about what actually happens in the cells? I thought it was like a mystery and there were so many things going on and we didn't really know. So how do drug discovery work now? Because it's just like molecule to protein, but there's an entire process before that so that the drug gets there and like
So Oh my God.
She's reading Danielle can have books, so that's why she has been very smart questions about all the interactions and about everything. So.
Yeah, so we don't know everything. There's most of the things that we don't know. And even if you talk with with researchers that work on the topic, they will be able to present you with a very well-defined picture of the things that they do know. And, you know, like everything else is blurred.
Right, yeah, so,
But then, you know, if you take 100 researchers working in different topics, and if you would be able to merge them, you would be able to get a better full picture of it.
no, but I'm asking like specifically here,
So specifically here,
one-to-one,
yeah,
and kind of neglecting the entire
Mhm.
environment of the cell. It's just like targeting one.
normally, yes. So normally they just, someone works in target identification and they just published, you know, this receptor, it's going to be very relevant for this
Yeah.
disease. And we have showed, you know, like a knockout or something like that, that this is going to be relevant. They don't know why. They don't know in some cases, they don't know the biological process or the cascades and everything that happens. But that is one of the first steps. And then two persons start to work in, one of them is trying to develop molecules that would bind there and try to inhibit or activate. And the other start to understand the chemistry by the walls.
And you are absolutely right, because one mentioned a fundamental thing that me and Joel have been discussing is you don't have it. So what you are saying is the clinical features that you have there on the patient side, they are not ready to the model. So you don't really know, you know, what is going to be the overall direction of the model. And selection is also always targeted to the thing that you would like to connect with. So that's a better.
But and also that's why most of the drug candidates fail, because then when they are going to validate them, you know, like in real or later stages, they did not took into account all of the other things that are happening.
Okay.
But maybe if we can incorporate, even if not everything, but everything that is already known somehow, computational model, we are able to test it. We are not there. Okay, I'm trying to be as clear as possible. But if in principle we are able to do that, then we are able to, you know, lower the failure rate.
Okay.
Because that is one of the biggest motivations from the big pharma. You know, if you spend 4 million or 40 million in a drug,
no, it's 1 billion
or 1 billion on a drug validation, and that you have to recruit patients, you have to stay with them, and then in the end fails. You'd like to increase as much as you can at the beginning in probability of success, because you know that will change the amount of models of the world are one of the fundamental parts of that. How to capture them, it needs to be, and that's why I don't think AGI is even close, because we cannot really capture all those together. But I'm curious about the images that you mentioned. So you mentioned that you are using images for the next step. You know, so you take the images and okay, and you are using convolutional neural networks or diffusion.
Diffusion has convolution.
Did you? So because there is Mahmoud on my back, and he's working on applying frequencies to image analysis. Okay. So would that be an experiment that you can run? So you have a heat map, okay? And if we run that heat map with images, the image, no, with frequencies, sorry, so not images, with frequencies. Frequency. Yeah. So if you shoot to them a range of certain frequencies that we know that behave in a certain way, for example, according to certain colors, would that be helpful?
I have no idea.
Okay.
Neither
do I. So we also don't have any idea if it's going to work, but we have just, we are just thinking, oh, this could be, what's, now because we tried, so.
I can tell the story. So my group, we are currently about 8 PhD students, because 2 of them, they are finishing. And because 2 of them are finishing. And this guy that it is here, it is a PhD student in my lab, and he was informatics student, bachelor, and biochemistry student at the same time when I met him. So he was my student in both courses. And then he went to data science, and I was able to motivate him to work with me. And before starting his PhD, he told me, look, you have something that you are doing really, really wrong, that you are using smiles and you are not considering, the real interference. And I want to work with this image representation. And I think that the best way is, to use this to represent the pharmacopoeias. Okay, he was already working with me for 2 years or 3 years, so we already have this. And I told him, if it wasn't you that were coming from these backgrounds, I have no interest in working with image because I don't have this. It has very specific, but let's give it a try. Okay, he has done an amazing work during his master thesis on this, but he failed in being able to show that it worked. Okay, but because he's very stubborn, now he's going to try it for the PhD.
Yes, so it's the online job. So to be a PhD student is to be stubborn because you have lots of things against it. And in the end, you get your PhD.
What is the type of image? Is it like an microscopy or?
No. So it is that for the hotspot, we take the heat map from
Well, I think that we will close the seminar.
Yes.
Yeah.
There is no
Thank you
questions.
very much.
I really want to thank you, all of you, for the questions and for the thinking that you were able.
Well,
I'm really impressed.
You will have probably more of them during your stay here. So thank you very much for your talk.
Okay,
Thank you so much.
Thank you very much for the discussion.